Hello, my dear students. Welcome to Sankalp Learning Classes. Today, we are discussing the important question that is from the evolution chapter, one of the important chapter for your board exam. So, already we have discussed the MCQs. In today's class, we are discussing the 2-mark questions as well as 3-mark questions. Okay. No 5-mark questions will be asked from this question for your board exam. So we are not discussing the 5-mark questions, only 2-mark questions and 3-mark questions we are discussing. Okay. Yes. First question. What are analogous organs? Give an example each from plants and animals. So you should write the definition of the analogous organs. Here, analogous organs so are the organs that have the different anatomical structure but they perform the similar functions. Means which are having the different anatomy but which are performing the same functions. For example, eye of the octopus and eye of the mammals, they are doing the same function. So that is the vision, alva. But they are that origin is different. Or the flippers of the penguins and dolphins. So both are performing the same functions but their origin is different. And one more example is bird wing and bird wing and butterfly wing. So that is also a example for the analogous organ. And one example, so that is from the plant, that is sweet potato and the potato. So both are doing the same function, that is the storage of the food, but their origin is different. Sweet potato, that is the root modification, and potato, that is the stem modification. So these are the some examples for the analogous organs. Same questions will be asked. What are homologous organs? Give examples. So what are homologous organs? Homologous organs, they have the similar anatomy, but they are performing the different functions. Same anatomical features will be there, but they are performing the different functions. For example, the thorns and tendrils of bougainvillea and cucurbita. Bougainvillea thorns, they are used for the protection, and cucurbita tendrils, they are used as a support. And one more example, whales, bats, and cheetah and human, all mammals, they share the similarities in the pattern of bones and forelimbs, but they perform the different functions. So in bats it is used for the flying, and in whales it is used for the swimming. So but they have the similar anatomical feature. They are called as a homologous organs. Next question. What is divergent evolution? Give an example. What is divergent evolution? So that is the definition. Divergent evolution is a process where species with a common ancestor develop different traits over time, eventually resulting in the development of the new species. For example, whales, bats, and cheetah, they have the similar anatomical features. Means they are evolved from the same ancestor. So according to this, but now they are performing the different functions. So same anatomical features are there, means they are originating from the same ancestor, but they are evolved to perform the different functions. So that is the example. Okay. Differentiate between the divergent evolution and convergent evolution. Differentiate, you have to write the difference between the divergent evolution and the convergent evolution. Here, divergent evolution, it involves the gradual separation of the species into two distinct species. This happens when the individuals in a species or closely related species acquire a variation in their traits. For example, whales, bats, humans, and cheetahs are share similarities in the pattern of bones and their forelimbs, but they are performing the different functions. Same anatomy, but they are performing the different function, that is the divergent evolution. And what is convergent evolution? So they are having the different anatomical features. It involves the development of similar traits into unrelated species. So this happens when the species occupy the similar ecological niches and adapt in a ways in response to similar selective pressures. For example, wings of insects, birds and bats are result in the convergent evolution, means their anatomy is different, but it is example for the convergent evolution. Next question. Give two examples of evolution by anthropogenic action, means that is human activities, due to the human activities. Here, industrial melanism is example for the anthropogenic evolution, and animals coloration or pattern changes blend into its environment. Industrial melanism, so one of the important topic. Illenaagತ್ತೆ ಅಂದ್ರೆ after the before the industrialization in England, the white moths are more compared to the dark winged moths. After the industrialization, so their number is reversed. Dark winged moths are more compared to the white wing. This is because before the industrialization, white winged moths, they are present on the bark of the trees which are having the lichens. They are the white color background ಅಲ್ಲಿ ಈ moths ಇರ್ತಿತ್ತು. So it is difficult to uh, identify by the predators, so they are survived. So these white color moths are more. After the industrialization ಏನಾಯ್ತು, lichens they are not found in the polluted areas alva. So the lichens are less, than the tree bark become dark, and in the contrast background white moths are identified by the predators, so the white moths are less. So this is example for the industrial melanism. And overuse of pesticides and weedicides. The overuse of these chemicals has led to the evolution of resistance varieties of pests and weeds. Some varieties are developed uh, by the overuse of the pesticides and the weedicides. Next question. Name any two theories that explain the origin of life. Here, how the organisms are developed, how the life is originated on the earth. So some theories are developed to explain this. The first theory, so that is the panspermia. So according to this, some spores from the outer planet that develop the new organisms in the on the earth, so that is the panspermia. And theory of spontaneous generation, it explains that from the dead or some non-living things, they are able to rise the they are from the non-living things, the living organisms are developed. From the non-living things, living organisms are developed. That is the theory of spontaneous generation. Theory of special creations means there is a special power, so we call it as a god. So God creates the all the organisms which are present on the earth. So these are the different theories, so which explains the origin of the life. Next question. How could the experiments of Louis Pasteur dismiss the theory of spontaneous generation? According to the spontaneous generation, ಏನಾಗಿತ್ತು? The non-living things, from the non-living things, the living organisms are arised. The first living organisms, they are arised from the non-living things. So that is called as a theory of spontaneous generation. But this theory was dismissed by the scientist Louis Pasteur by his experiment. But what is that experiment? Louis Pasteur, by careful experimentation, demonstrated that life comes only from the pre-existing life. He showed that pre-sterilized flask, then life did not come from the killed yeast while it another flask opened to air. New living organisms arose from the killed yeast. So spontaneous generation, so that is dismissed by this scientist. So what he has done, he has taken the two flask, sterilized flask. So in one flask, he kept open flask, so which is having the killed yeast. In the another flask, it is closed and killed yeast are kept inside this, but the flask which contain the sterilized so which contain the killed yeast and where the which is not open, so that does not contain any where no organisms are developed in this, but which is kept open, so where the organisms are developed. So he told that, so according to this experiment, so he showed that there is no living organisms developed from the non-living things. Next question. State the evolutionary relationship between giving reasons between the thorns of bougainvillea and tendrils of cucurbita. Here, the bougainvillea thorns and cucurbita, here, tendrils of the cucurbita, they have the similar origin. Origin is same, but they perform the different functions. Thorns of the bougainvillea they used for the protection, but cucurbita, that is the tendrils of the cucurbita, that is used for the support. Here, both are originate from the exile of the leaf, but they are anatomically similar but performs the different functions. So they are called as homologous organs, and they show the divergent evolution. They show the divergent evolution. They showed the divergent evolution. Next question. Select the homologous structure from the combinations given below. Which are the following are the homologous organs here? Tuber of the potato and sweet potato, they are analogous organs. Analogous organs because they have the same different origin. Tuber of the potato, that is the stem modification. Sweet potato, that is the root modification, but performs the same function. Salva, because of that they are the analogous organs. Eyes of the octopus and eyes of the mammals, they are also analogous organs because they perform the same functions. Forelimbs of whales and bats, they are called as homologous organs. Why they are homologous organs? Thorns of bougainvillea and tendrils of cucurbita, their origin is same, their anatomy is same, means their originate is same but performs the different functions. These also are homologous organs. Hagadre illi 3 and 4, they are the homologous organs. Why they are homologous organs, that also you have to mention. And tuber of the potato and eyes of the octopus, they are the analogous organs. I will write A here. Why they are analogous organs, that also you should write. Next question. Mention the two features of the following: Ramapithecus and Australopithecus. You have to write the two features of these humans. What are the characteristics of the Ramapithecus? Ramapithecus have the hairy body and upright walking. They are not walking like a chimpanzees or apes. So they are more man-like. They are having the more man-like structures. And Australopithecus, so the evidence shows that they hunted with stone weapons. They used the stone weapons and they ate the fruit. Essentially, they they ate the fruits. So that is the characteristics of the Australopithecus and Ramapithecus. ಇಲ್ಲಿ ನಿಮಗೆ write the features of Homo habilis ಕೇಳ್ಬಹುದು. So just I'm giving the example. Homo habilis ಕೇಳ್ಬಹುದು. Homo erectus, Homo erectus, Neanderthal man ಕೇಳ್ಬಹುದು. So so you have to practice the features of these all humans. Homo erectus human evolution steps ಏನಿದೆ ಅಲ್ವಾ ಅಲ್ಲಿ ಎಲ್ಲ ಒಂದು ಫೀಚರ್ಸ್ ಅನ್ನ ಪ್ರಾಕ್ಟೀಸ್ ಮಾಡ್ಕೊಂಡಿರಿ. ಯಾವುದು ಕೇಳ್ಬಹುದು. ಹಿಯರ್ ಐ ಹ್ಯಾವ್ ಟೇಕನ್ ದ ರಾಮಾಪಿಥಿಕಸ್ ಅಂಡ್ ಆಸ್ಟ್ರಲೋಪಿಥಿಕಸ್. Next question. How does Miller's experiment supports the theory of chemical evolution? So Miller's experiment, that is one of the very important experiment for your exam. Here, formation of diverse organic molecules from the inorganic constituents. So that is the theory of chemical evolution. The conditions on the early earth was high temperature, volcanic storms, reducing atmosphere containing the CH4 ammonia, methane, ammonia, etc. So for this experiment, Stanley Miller, he created the similar condition in the laboratory scale. He created the electric discharge in a closed flask containing CH4, H2, hydrogen and ammonia and water vapor at 800 degrees Celsius. Then what he observed? He created this condition in the laboratory scale. Then what he observed? He observed the formation of amino acids in similar experiments. Others observed formation of the sugars, nitrogen bases, pigments and fats. So the analysis of the meteorite content also revealed that similar compounds, indicating that the similar processes are occurring elsewhere in the space. Means same happened on the earth. So the experimentally it is proved by the Stanley Miller. What is genetic equilibrium and founder effect? So you have to define for each 1 marks what is genetic equilibrium and founder effect. Here, genetic equilibrium is a theoretical state where the frequency of alleles or genotypes in a gene pool remains the same from generation to the generation. That is the Hardy-Weinberg equilibrium. The genetic equilibrium is a theoretical statement that where the frequency of alleles or genotypes in a gene pool remains the same from generation to generation. Then what is founder effect? The original drifted population becomes founders and effect is called. When the drifted population becomes the founders in the new place. Alva. So they are called as, that is called as a founder effect. So these are the some 2-mark questions what we have discussed. So next we are discussing the some important 3-mark questions. Sketch and label the Miller's experiment and explain it. So ಈ ತರನು ಕೂಡ ಕೇಳ್ತಾರೆ. ಈ diagram is very important diagram. ಇದನ್ನ ನಿಮ್ಗೆ ಡಯಾಗ್ರಾಮ್ ಬಿಡಿಸ್ಲಿಕ್ಕೂ ಕೂಡ ಕೇಳ್ತಾರೆ ಕೆಲವೊಂದ್ಸಲ. ಎಕ್ಸ್ಪ್ಲೇನ್ ದ ಸ್ಟ್ಯಾನ್ಲಿ ಮಿಲ್ಲರ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಅಂತಾನು ಕೂಡ ಕೇಳ್ತಾರೆ. ಲೇಬ್ಲಿಂಗ್ಸ್ ಅನ್ನ ಕರೆಕ್ಟ್ ಆಗಿ ಮಾಡಿ. ಸೊ electrodes, then what are the gases used, then water out and water in and boiling water, vacuum pump. So these all labelings you have to label properly. ಇದನ್ನ ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಆಗಿ ಕೇಳ್ತಾರೆ as a 3-mark or 2-mark question ಅಲ್ಲಿ ಕೇಳ್ತಾರೆ. And one more important thing, you have to label properly. Correct agi label ಮಾಡಬೇಕು. And explanation also sometimes they may ask. Explanation ಕೂಡ ಕೆಲವೊಂದ್ಸಲ ಕೇಳಬಹುದು. Then next question. What is adaptive radiation? Explain it with reference to the Darwin pinches. Yes, what is adaptive radiation? Here, so this is the process of evolution of different species in a geographical area, starting from a point and literally radiating to the areas of the geography is called as a adaptive radiation. Means adaptive radiation, it means, so there is a single ancestor. Just imagine this is the ancestor, so which is present in a particular area. When these organisms, they will move to the different region of that different regions, according to that regions they will modify their body structures, or they will adapt. For that adaptation they will modify, they will change, they will evolve. So that is called as a adaptive radiation. Here Darwin pinches is a one of the good example for the adaptive radiation. ಇಲ್ಲಿ ನೋಡಿ, Darwin realized that there were many varieties of pinchers in the same island. So there are different varieties of pinchers observed in the same island, and all the varieties he conjectured evolved on the same island itself. Means they are evolved from the same ancestor. From the original seed eating features, so that is the original is seed eating features, and then many other forms with altered beaks arose, enabling them to become a insectivorous and vegetarian pinchers. So first ಯಾವ ತರ pinchers ಇತ್ತು, original is the seed eating features. When it is moved to the different areas of that island, they are adapted for that condition. So they become that is the pinchers are modified into insectivorous and some are vegetarian pinchers. So like this they are evolved. So that is the example for the adaptive radiation. Next question. Mention the different stages of human evolution in a correct sequence. In a correct sequence, you have to mention the different stages. This is the 3-mark question. Dryopithecus. So he's like more ape-like character. He's having the more ape-like character, hairy body, and Ramapithecus having the more man-like character. So he is more man-like character. Then Australopithecus, they use the uh, stones, and they mainly they ate the fruits. Then Homo habilis, Homo habilis. So these are the first man-like uh, man-like, which are Homo habilis, which is having the more man-like character. Then Homo erectus, their brain capacity, and Neanderthal man brain capacity is 1400 cc. And what are the features of the Neanderthal man? That is also very important. Then just 10,000 years ago, Homo sapiens are appeared. So this is the correct sequence of this human evolution. Very important, each stage, characteristics of these characters. Then brain capacity, so which it, if it is given brain capacity irbodu, when they evolved, when they found, means 15 million years ago, or 2 million year ago ಆ ತರ ಕೊಟ್ಟಿದ್ದಾರೆ. Then what are the features, like there are different features where they evolved. For example, in which part of the earth, like for example Asia, some are found in Asia, like that. So ಇದನ್ನೆಲ್ಲ ಒಂದು ಪಾಯಿಂಟ್ಸ್ ಮಾಡಿ ಪ್ರತಿಯೊಂದಕ್ಕೂ ಫೀಚರ್ಸ್ ಅನ್ನ ಬರ್ಕೊಳ್ಳಿ. ಸೊ ಈ ಎಲ್ಲ ಪಾಯಿಂಟ್ಸ್ ರಿಲೇಟೆಡ್ ಪ್ರತಿಯೊಂದು ಏನ್ ಕೇಳಿದ್ದಾರೆ ಅಲ್ವಾ, ಟ್ರಯೋಪಿಥಿಕಸ್, ರಮಾಪಿಥಿಕಸ್, ಅದ್ರ ಬಗ್ಗೆ ನೀವು ಪಾಯಿಂಟ್ಸ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಅದು ನಿಮ್ಗೆ ತುಂಬಾ ಹೆಲ್ಪ್ ಆಗತ್ತೆ, ಇವನ್ MCQ ಆಸ್ ವೆಲ್ ಆಸ್ ಫಾರ್ ಯುವರ್ ಲಾಂಗ್ ಆನ್ಸರ್ ಕ್ವಶನ್. Next question. State Hardy-Weinberg law. List 4 evolutionary factors which disturb genetic equilibrium. The Hardy-Weinberg principle states that genetic variation in a population will remain constant. So that genetic variation remain constant from generation to generation if there are no disturbing factors. If there are no disturbing factors, then only the genetic variation in a population remain constant. Now, what are the factors which affect this? First one, mutation, genetic drift, natural selection, genetic recombination, and gene flow. So these are the different factors which affect the Hardy-Weinberg principle. Define the terms saltation, gene pool, and genetic drift. So you have to define these three terms. What is saltation? What is gene pool? And what is genetic drift? Here, what is genetic drift? Genetic drift is the change in a frequency of an existing gene variant in the population due to the random chance. So the genetic drift is the change in the frequency of an existing gene variant in the population due to the random chance. Next question. The gene pool. What is gene pool? The gene pool is the collection of all genes and alleles in a population of a species. The gene pool that refers to the collection of all the genes and alleles in a population of a particular species. Next question. What is saltation? Saltation is a biological term that refers to a sudden large scale mutation that can occur in a population from one generation to the next. So that is the sudden mutation, sudden large scale mutation. In a simple words, what we can say, sudden large scale mutation is called as a saltation. Next question. How paleontological evidences have helped understanding the evolution of life forms. Fossils are remains of the hard parts of life forms found in the rocks, and rocks form sediments, and cross section of earth crust indicate the arrangement of the sediments one over the other during the long history of earth. So here different aged rocks, when you observe the different aged rock sediments contain the fossils of the different life forms who are probably died during that formation of the particular sediment. So the organisms which are found in the sediments, so they represent the organisms lived in that era, and some of them appear similar to the modern organisms. They represents the extinct organisms, for example, dinosaurs. A study of fossils in different sedimentary layers indicate the geological period in which they existed, when they are existed, so that is represented. And the study showed that life forms varied over the time, and certain life forms are restricted to a certain geological time spans. Hence, the new forms of the life have arisen at different times of the history of earth. All these are called as a paleontological evidence. Means the different organisms, they are evolved in the different era, and some are extincted and some are evolved, some are changed, adapted for the new environmental conditions. So these all things, so they are seen by observing the sediments which are the organisms which are buried under the sediments. Next question. What were the different gases did the flask used an experiment set up by the Stanley Miller contained? So ಯಾವ gases ಅನ್ನ ಯೂಸ್ ಮಾಡಿದ್ರು ಫ್ಲಾಸ್ಕ್ ಅಲ್ಲಿ? CH4, methane, ammonia, water and hydrogen is used. Then what conditions of primitive earth was recreated in the flask? So ಯಾವ conditions ಅನ್ನ create ಮಾಡಿದ್ರು? The condition on earth were high temperature and reducing atmosphere contain the ammonia, methane, etc. So that is the condition which was created in the laboratory scale. And write the conclusion drawn from this experiment. In similar experiments, others observe formation of the sugars, nitrogen, bases, pigment, and fats. So formation of the some contents, and analysis of meteorite content also revealed that similar compounds, indicating that the similar processes are occurring elsewhere in the space. Next question. After industrialization in England, it was observed that white-winged moths not survive. White-winged moths survive ಆಗ್ಲಿಲ್ಲ after industrialization. ಯಾಕೆ? What is the cause? ಯಾಕೆ white-winged moths numbers ಏನಾಯ್ತು ಕಡಿಮೆ ಆಯ್ತು? Because during the post-industrialization period, the tree trunks became dark due to the industrial smoke and soot. A tree trunks ಏನಾಯ್ತು black color ಆಯ್ತು. Under this condition, white-winged moths did not survive. So, ಆ ಒಂದು ಟ್ರೀ ಟ್ರಂಕ್ ಕಲರ್ ಬ್ಲ್ಯಾಕ್ ಇತ್ತು, ವೈಟ್ ಮಾತ್ಸ್ ಇತ್ತು. ಸೊ ದೇ ಆರ್ ಈಸಿಲಿ ಐಡೆಂಟಿಫೈಡ್ ಬೈ ದ ಪ್ರಿಡೇಟರ್ಸ್. ಸೊ ದೇ ಆರ್ not they, but the dark winged or melanized moths were survived. Black color background ಅಲ್ಲಿ black color or dark color wing ಇರುವಂತಹ moths ಏನಾಯ್ತು survive ಆಯ್ತು. White color they are not survived. What was the change and why it happened? What was the change? Sorry. Before industrialization set in, the thick growth of almost white color lichens. ವೈಟ್ ಕಲರ್ ಲೈಕೆನ್ಸ್ ಏನಾಗಿತ್ತು ಆ ಒಂದು ಟ್ರೀ ಟ್ರಂಕ್ ಮೇಲೆ ಇತ್ತು. ವೈಟ್ ಕಲರ್ ಬ್ಯಾಕ್ ಗ್ರೌಂಡ್ ಅಲ್ಲಿ ವೈಟ್ ಕಲರ್ ಮಾತ್ಸ್ ಏನಾಗಿತ್ತು, ವೈಟ್ ವಿಂಗ್ಡ್ ಮಾತ್ಸ್ ಸರ್ವೈವ್ಡ್. ಬಟ್ ಡಾರ್ಕ್ ಕಲರ್ ಮಾತ್ಸ್ ವರ್ ಪಿಕ್ಡ್ ಔಟ್ ಬೈ ದ ಪ್ರಿಡೇಟರ್ಸ್. ವೈಟ್ ಕಲರ್ ಬ್ಯಾಕ್ ಗ್ರೌಂಡ್ ಅಲ್ಲಿ ಬ್ಲಾಕ್ ಕಲರ್ ಆರ್ ಡಾರ್ಕ್ ವಿಂಗ್ಡ್ ಮಾತ್ಸ್ ಇದ್ರೆ ಏನಾಯ್ತು, ಅದು ಪ್ರಿಡೇಟರ್ಸ್ ಇಂದ ಕ್ಯಾಪ್ಚರ್ ಆಗ್ತಿತ್ತು ಅಲ್ವಾ. ಸೊ ದಟ್ ಈಸ್ ಬಿಫೋರ್ ದ ಇಂಡಸ್ಟ್ರಿಯಲೈಸೇಷನ್. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. Which organism is known as natural indicator to air pollution? So that is lichens. Lichens grow where the air grow on the tree trunk where the air pollution is less. Air pollution ಕಡಿಮೆ ಇದ್ದಾಗ ಆ ಬಾರ್ಕ್ ಮೇಲೆ ಏನಾಗ್ತಿತ್ತು, ವೈಟ್ ಕಲರ್ ಲೈಕೆನ್ಸ್ ಗ್ರೋ ಆಗ್ತಿತ್ತು. ಬಟ್ ಆಫ್ಟರ್ ದ ಇಂಡಸ್ಟ್ರಿಯಲೈಸೇಷನ್ ಏನಾಯ್ತು, ಏರ್ ಪೊಲ್ಯೂಷನ್ ಜಾಸ್ತಿ ಆಯ್ತು, ಲೈಕೆನ್ಸ್ ದೇ ಆರ್ ನಾಟ್ ಗ್ರೋಯಿಂಗ್ ಅಂಡ್ ದ ಟ್ರೀ ಟ್ರಂಕ್ ಕಲರ್ is also changed. So that is example for the industrial melanism, one of the important topic. Or they may ask, write about the what is industrial melanism, explain it with reference to the white winged moths and the dark winged moths. ಈ ತರನು ಕೂಡ ನಿಮಗೆ ಕೇಳ್ತಾರೆ. Next question. What is mutation? Who proposed the idea of mutations? Here a mutation is a change in the DNA sequence and organism's genome. Mutation can be caused by errors during the cell division or exposure to a DNA damaging agents or other types of the damage to the DNA. So this mutation, so this we already studied, we are familiar with this word mutation, and this is proposed by the scientist Hugo de Vries. The mutation is studied by the scientist Hugo de Vries. Next question. Explain the adaptive radiation with suitable example. Explain the adaptive radiation anna ನಾವು we have seen in the Darwin pinches ಅಲ್ಲಿ ನೋಡಿದ್ದೇವೆ ಅಲ್ವಾ. Darwin pinches ಅಲ್ಲಿ ನೋಡಿದ್ದೇವೆ. You can use one more example, that is Australian marsupials. I will take the example Australian marsupials. ಇಲ್ಲಿ ಎನಿ ಎಕ್ಸಾಂಪಲ್ ಸುಟೇಬಲ್ ಎಕ್ಸಾಂಪಲ್ ಅಂತ ಕೊಟ್ಟಿದ್ರೆ ನೀವು ಡಾರ್ವಿನ್ ಪಿಂಚಸ್ ತೆಗೆದುಕೊಳ್ಬಹುದು, ಆಸ್ಟ್ರೇಲಿಯನ್ ಮಾರ್ಸುಪಿಯಲ್ಸ್ ತೆಗೆದುಕೊಳ್ಬಹುದು. ನಾನಿಲ್ಲಿ ಆಸ್ಟ್ರೇಲಿಯನ್ ಮಾರ್ಸುಪಿಯಲ್ಸ್ ಎಕ್ಸಾಂಪಲ್ ತೆಗೆದುಕೊಳ್ತಾ ಇದ್ದೇನೆ. ಸೊ ವಾಟ್ ಈಸ್ ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್, ದಟ್ ಯು ಹ್ಯಾವ್ ಟು ರೈಟ್. Here, Australian marsupials, a number of marsupial, each different from other, evolved from the ancestral stock, but all within the Australian island. So these are the marsupials, but within the Australian island, they are evolved. When more than one adaptive radiation appeared to have occurred in an isolated geographical area, so one can call this as a convergent evolution. So one more example is placental mammals in Australia also exhibit the adaptive radiation. So evolving into varieties of such placental mammals, each of which appears to be a similar to corresponding marsupial. So that is the placental wolf or a marsupial. So this is example for the ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್. ನೀವು ಯಾವುದೇ ಎಕ್ಸಾಂಪಲ್ ತೆಗೆದುಕೊಳ್ಬಹುದು, ಆಸ್ಟ್ರೇಲಿಯನ್ ಮಾರ್ಸುಪಿಯಲ್ಸ್ ತೆಗೆದುಬಹುದು ಅಥವಾ ಪ್ಲಾಸೆಂಟಲ್ ಮ್ಯಾಮಲ್ಸ್ ಅದನ್ನು ಕೂಡ ತೆಗೆದುಕೊಂಡು ಆನ್ಸರ್ ಅನ್ನ ಬರಿಬಹುದು. ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್, ರೈಟ್ ದ ಫೀಚರ್ಸ್ ಆಫ್ ಹೋಮೋ ಹ್ಯಾಬಿಲಸ್ ಅಂಡ್ ನಿಯಂಟ್ರತಲ್ ಮ್ಯಾನ್. ನಿಯಂಟ್ರತಲ್ ಮ್ಯಾನ್ ಮತ್ತೆ ಹೋಮೋ ಹ್ಯಾಬಿಲಸ್ ಫೀಚರ್ಸ್ ಅನ್ನ ಬರಿಬೇಕು ನೋಡಿ. Homo habilis, this creature was called the first human-like being. The hominid was called Homo habilis. Here, what are the brain capacity of the Homo habilis? About 650 to 800 cc. They probably did not eat meat. So this is the feature of the Homo habilis. So the first human-like being, so that is called as a Homo habilis, and brain capacities were between the 650 to 800 cc. The brain capacity is about 650 to 800 cc and probably did not eat meat. So these are the characteristics. Next, Neanderthal man. Neanderthal man with a brain size 1400 cc lived in near East and Central Asia between, so these years, around 1 lakh to 40,000 years back. And they used hides to protect their body and buried their dead. So these are the features of the Neanderthal man. So they may ask like this, write the name where the man found near the Central Asia and near East and Central Asia between 1 lakh to 40,000 years back. So that you have to write Neanderthal man. ಈ ತರನು ಕೂಡ ಕ್ವಶನ್ಸ್ ಅನ್ನ ಕೇಳ್ತಾರೆ. Okay. So these are the some questions, 3 mark questions related to this chapter. So here I will end the class. So I hope these all questions useful for your board exam. So these are the very important questions. So please practice these all questions. So this will be really helpful for your board exam. Thank you.